In this video, we will learn about fine structure of chromosome. Now, chromatin material condenses to form chromosomes when the nucleus enters into cell division. Chromatin is formed of two substances, deoxyribonucleic acid, that is DNA, and the basic proteins called histones. Deoxyribonucleic acid or DNA. DNA forms about 40% while histones form about 60% of the chromatin. DNA and histones form a kind of complex structure in which histones form small core particles and DNA is wrapped around these core particles, forming the nucleosome. Each nucleosome has one core particle formed of 8 molecules of histones. DNA makes 2.5 coils around it. Thus, an interface chromatin fiber has numerous nucleosomes which are arranged like beads on a string. Such a chromatin fiber coils and supercoils to form thick metaphase chromosome. So here we can see that this is the histone protein forming core particles and 8 histone proteins are there. Then this is the DNA wrapping the core particles and together they are forming the nucleosomes. This is the double helical structure of DNA. Such numerous nucleosomes are forming the chromatin fibers and they actually form the chromatids of chromosomes. Structure of DNA. DNA is a macromolecule formed of two polynucleotide strands or chains. They are complementary and antiparallel to each other. They are spirally coiled on each other. Each polynucleotide strand is composed of thousands of nucleotides. Nucleotides are structural units of DNA. They are called monomers. Each nucleotide is formed of three components, namely a nitrogenous base, a molecule of pentose sugar, or that is deoxyribose in this case, and a molecule of phosphate. The nucleotides in polynucleotide chain are arranged in such a way that the phosphate and sugar molecules are arranged lengthwise alternately. The nitrogenous base are attached to the sugar molecule and are laid horizontally inwards. The nitrogenous bases of nucleotides of two polynucleotide chains are joined together through hydrogen bonds. The nitrogenous bases found in DNA. The four types of nitrogenous bases are found in DNA. These are adenine, guanine, thymine, and cytosine. Always adenine pairs with thymine and guanine pairs with cytosine. That is A forming double hydrogen bonds with T and G forming triple hydrogen bonds with C. Due to this pairing, the two strands are complementary to each other. The sugar-phosphate chains form sides of a ladder and nitrogenous base pairs from the rungs. This arrangement of sugar, phosphate, and nitrogenous bases give a spiral ladder-like shape to the DNA molecule called double helix and forms double helical structure of DNA. So here if we see that the sugar, that is a pentose sugar called deoxyribose, and it combines with the base, and these two are actually nucleosides. And when it is connected to a phosphate group, then the entire structure is known as nucleotide. Then we can see that the cytosine is paired with guanine with 3 hydrogen bonds and adenine is paired with thymine with 2 hydrogen bonds. So the nitrogenous bases of two strands are paired among themselves to form the double helical structure of DNA. And we can see that these strands are complementary and anti-parallel to each other. I hope this topic is clear to you. Thank you.